Welcome everybody to the 34th edition of the Multi-Omics Reading Group. I'm your host for today. And today, after very many discussion sessions that Professor Romain Lopez, who is now presenting at the Reading Group, let us in on, we have the pleasure of having him host a talk right here at this Reading Group. So before we actually go forward with that, Sinu, please say your wonderful introduction as you often do.
Yeah, thanks, Cesar, for the nice beginning. Okay, hi everyone. Welcome Romain Lopez, who is our speaker today. So Romain is an assistant professor at computer science and biology at New York University. He received his master's in applied mathematics from Ecole Polytechnique and his PhD in electrical engineering and computer science from UC Berkeley, advised by Professor Michael Jordan and Niels Joseph. He was postdoc fellows at Genentech and Stanford University, hosted by Professors Aviv Regev and also Jonathan Pichard. He has received best paper honors at leading AI and computational biology venues, as well as STAT Winterkind Award in 2024, which is very amazing. He also is recognized by the North America's most promising early career scientists. His research develops probabilistic and causal machine learning methods to uncover the mechanism that govern cellular behaviors and disease, which also include the SCVI framework, which is super popular, and also the SCVI tools ecosystems for the deep generative modeling for single-cell omics data. So let's welcome Romain Lopez. Thank you for accepting our invitation, and it's all yours. Thanks.
Thank you. Thank you very much for inviting me today. And I'm very delighted to be here and to present about some of our recent work on cellular dynamics. So thinking about how cells respond through time and also how they respond to perturbation. And so I will be presenting one line of work, and I hope that I'll be able to work a lot more in this specific type of problems. So I will start about talking about the group that so I started at NYU actually a month and a half ago, so this is still pretty new. I created this biological machine learning group sitting in between biology and computer science, and I would like to use this opportunity to describe the interest of the group. So the group is very interested in something we're going to talk about today, which is models of cellular dynamics. Thinking about how cells respond to a signal or stimuli through time, and how that helps understand how we can understand the mechanism behind this behavior. I'm interested in T-cell biology and applying some of the modeling tools I will talk about today to T-cells. And T-cells actually are a very important part, and they were actually T-cell discoveries were used this year to receive the Nobel Prize in medicine. I will also talk about, I'm also interested in causal models for perturbation data and thinking about it also in the context of multiple modalities. And one last area that's dear to the lab is sometimes you cannot run all the experiments you would like. And in that setting, perhaps having some machine learning model that can help with optimal experimental designs could be pretty helpful. And so this is some kind of work that the lab is started focusing on as we're just started. But this is a non-exhaustive list. I'm interested in a lot of intersection between cellular biology and machine learning. And there is something strange. Okay. So and today we will focus on the intersection of models of cellular dynamics, and I will motivate this from a T-cell biology perspective. So this is joint work. The work I will actually present today is the scope of a single paper, and that paper was actually an intern's project that I was supervising while I was at Genentech. And so the intern was Martin, who did an amazing work visiting from Heidelberg and and was at Genentech for the summer. This was a great collaborative work where we got the involvement of a few people who had been working on optimal transport and those dynamical systems type of work for a while. So Edward, Charlotte, and also Jean Christian. And this was co-supervised with Aviv Regev. So I will start by giving a brief background on T-cell biology and to try to motivate the problem. So T-cells, and specifically T-cell activation, is a central piece of the immune response. So here is a T-cell, very caricatured, and the T-cell might encounter an antigen. The antigen is presented by another cell, and essentially when this kind of this contact happens, we call that T equals 0. This is the beginning of T-cell activation and the T-cell response. And what happens is this is the start of a lot of beautiful changes in the immune system. So that T-cell will divide and expand. It's called clonal expansion, and that will help with having many cells able to recognize antigen. Those T-cells will then migrate to the affected areas, and depending on there are different types of T-cells. Some of them will be able to destruct infected cells, for example, by a virus, and some other cells will be able to recruit other immune cells or signal to them so that the immune system can really start working. So, and again, this is what, for the purpose of modeling dynamics, we will refer to that specific event of T-cell activation at the beginning is like T equals 0. So I'm mentioning this T equals 0. Okay. I'm mentioning this T equals 0 because T-cell activation is temporally structured, which makes it a beautiful system for thinking about dynamics. For example, what people have done is looking at plenty of gene expression data at many time points and trying to understand whether we can nominate groups of genes or gene programs that are specific to certain time points. And again, we start at T equals 0 here, which is the activation. And by doing some data analysis, you can kind of recover, here it's 4 gene programs that seem to be transient, right? So some of those genes are basically activated at certain time points, and some of them shut down later on, and other activates. And what we can essentially see is that at the very beginning, we have high activation of some marker genes for naive state, which makes sense. The T cells are resting, and it takes a while for gene expression to change. After a few hours, we already see activation of a bunch of genes related to calcium intake and some early signaling that then moves onto production of certain cytokines that are helpful for the cells proliferating. And then we finally see all those genes related to cellular proliferation. So again, biology has a lot of like beautiful dynamical systems or processes, and here is an example of one of them. And T-cell is specifically exciting because we use in the clinic small molecules or drugs to somehow modulate T-cell activation. And so I'm going to talk to you about 2 specific drugs and their example of clinical usage. So for example, there is this drug called dasatinib, and I put the chemical structure here. I'm not a chemist, so actually I can't read those really well. I would love to learn a bit more about that. But so dasatinib is a drug that you can use, and we know that it targets a very early step of activation, and it essentially acts as an off switch, meaning that the cells are almost not activated anymore. And this is useful. It has been approved for usage in leukemia, and it's also used experimentally for some of the engineered T-cell therapies that are going on now, for example, for cancer and controlling their toxicity. Another kind of drug that functions differently is solsitinib. And so that drug, basically, we know it targets a much later step of activation, a few hours later. And so what it does is it cannot do the same thing that the first drug, right? If it acts later, it induces a partial response, and essentially it will reduce the inflammatory response. And so this is very helpful for autoimmune diseases where immune cells can attack your own body. So like psoriasis, for example. So that specific drug is used in research, and other similar drugs have been approved for those autoimmune diseases. Okay. So we've seen the dynamical systems, and we've seen ways to perturb the system. And so this brings to data, right? So we're going from biology to data, and then we'll jump into the machine learning problem. So this is a kind of data that you can work with that allows you to understand the effect of drugs like interventions and profiling with some kind of dynamics, okay? And so here I'm going to talk about single cell sequencing, which is, this is a multi-omics reading group. So now we're a bit more on topic. So this is a collaboration with Professor Kelvin Chen from Osaka University. We've been close collaborators for a few years and thinking about those chemical screening data and more recently thinking about time points. So the experimental framework works as follows. We can take some CD4 T cells, we can plate them, so and we can profile around 500 drugs, which is a pretty big screen for this kind of technology. I'm showing you here one plate, but you need to pile up like probably more than around 30 or more plates for getting the kind of scale that you need. We can do this drug screen at different dosages. We activate the T cells synthetically using those antibodies. There is no antigen here. And then we basically profile with single cell RNA sequencing, and we can repeat this at multiple time points in parallel, okay? So for those of you who don't know what the data looks like, I will summarize it very briefly. Oh, this is really not working today. All right, so we have for every single cell, so those are the rows, and we can have up to millions of cells. We measure the expression of genes, for example, tens of thousands of genes, and these are usually count matrices that we can process. And then we also know for every cell, this is strange, which time point it came from, as well as the drug and the dosage that was used for the perturbation, okay? And so this is the data we will work with. Again, many cells, each cell comes from a single time point and a single perturbation, okay? So from this kind of data, we can be interested in biological questions, okay? For example, how do this drug affect T cell activation? In which magnitude, at which time point? We can try to understand whether the drug effects are consistent with the documented target. So each of those drugs are supposed to be targeting a certain, let's say, a kinase, and we can try to see if this is the case or not. We can also try to understand, because here we have dosages, so chemistry is actually quite complicated here, whether at high or low dosages, the effect can be very different and for which reason. And these are not the questions I will talk about today, but these are very important starting points for somehow reasoning about this data and think about what kind of model could be helpful in making meaningful contributions to computational biology. So now that I've talked a little bit about the whole context for why I'm interested in perturbation and dynamics, I kind of want to narrow down the focus for this talk. About addressing a particular challenge that comes up with those temporal measurements. So the idea is that the cost of such experiments is linear in the number of drugs and the number of time points. And for computer scientists, linear is not expensive, but with the reality of price of experiments in the lab, it's already it is expensive. So we would like to perhaps think about a way of scaling by profiling different. We don't need to do all the drugs in all the time points, okay? If we have decent overlap, so for example, something like this, where some of the drugs are profiled at some time points and some of the drugs at some other time points, we would be able to somehow impute the missing data, okay, or the missing distribution. And so this is kind of what we want to do, and this is the problem that I will focus on for today. I will present a flow matching framework for somehow making these experiments more efficient and for inferring the distribution of the single cell data at these missing combinations of time and drug, okay? And the idea is that this is a reasonable thing to do. This is because many drugs are expected to have similar effects at the same time points. Okay, and I will finish on this introduction of the biology on this point. I was having conversations here with people earlier, and one of the topics that came up was about biology and out of sample generalization. And so I want to highlight that for this particular problem somehow, because every drug somehow is profiled in at least one time point, this is not a hard problem of completely out of sample generalization. And if we have a model that can fit the dynamics, we can hope that this is somehow a solvable problem. Okay. All right. And so now I would like to just stop here quickly and see if there are any questions on the setup and the problem we're trying to answer.
I guess I have a question about the different time points at which different drugs operate, right? You had a slide comparing two drugs, right? So would one goal also to kind of do the sort of reverse inference of kind of like identifying key time points at which we would want a drug to operate on? And try to design the drug. I mean, it's a bit in the other way around, but I wonder if that's
good. That's good, right? So So those are kind of like completing these problems that complete each other, right? So either you haven't done any experiments and you're trying to understand, okay, I have all of these molecules and all of those time points. Maybe let's say knowing which time point, you do need to bring in prior knowledge, like things I've showed you before, right? So you already know which time points you want to do, for example, and then you might want to reason like, if you have an embedding of the molecules, for example, you kind of want to cluster that and take some diversity sampling, and then you want to take, you want to make sure that you cover the space basically. And so then some of the small molecules will be done at a few time points just to know which small molecule they cluster with. And some other molecules will have to be profiled at many, many time points such that you get some kind of breadth, right? So you can think about doing that in those two modes in some kind of greedy way. You can think about a machine learning method for solving that problem with some data. So that would be some kind of active learning problem. But in this setting, I guess I'm interested in, let's say we have a design, and the design looks like something like this. And we want to, once we have the design, we want to input the data.
Right.
Yeah, but this is a great question. Yeah, thank you. Thank you. Okay. All right. So I will move on. And so now I would like to give a brief background on those generative models and on flow matching. So let's have a look at how flow matching works. So in flow matching, I'm interested in an input distribution, or like I guess a distribution P0 that is essentially formulated as a noise distribution. So for example, here we have a noisy image, and we would like to somehow find a way to link it to a distribution over data points, or distribution of natural images. And it seems like the task is not super easy, but with the way that flow matching works is that you introduce some kind of a time variable. And I want to highlight that the time in flow matching has nothing to do with the experimental time, at least in this form. So there is a fictitious time somehow, and there is a function that we will see. This is a vector field that somehow, maybe for every particle in the noise, we can learn how to map it to a given image. Okay. And if we can do that somehow when we start from the noise and we integrate this vector field, I will specify this more carefully, there is some kind of dynamic process that helps us restoring or like sampling natural images, like the image of the cat here. So we need to specify a little bit more of like the mathematical tooling that is used in flow matching. Again, we're going to need to use those vector fields. So what is a vector field? It is a function that takes as input the time and the space, and that outputs a vector in the space, but that vector is actually somehow intuitively the derivative of, at this specific point in time, t, if we start from x and we wait time dt, like where are we moving, right? This is kind of the derivative. So this is how the mathematical object, the vector field, if we start at a given time point x and we follow the vector field and we integrate this differential equation, we obtain the flow. So a flow is a solution of this differential equation, and it tells you how the particle moves from time 0 to any point t starting from x at time 0. Okay, it's obvious how those two objects are related. And then there is a probability density path. So a probability density path is how, when you start from a distribution P0, if you push forward with the flow, so you push each particle basically, you end up with a transformed probability density. And as a function of time, this becomes a path, and you follow all the density and how the densities are changed through time. Okay. So, and now when I'm writing this, going back to this framework, this makes a little bit more sense. So we have this, we want to integrate, we want to start from a particle, we want to integrate and end up with an image of a cat, and that's how the denoising works. And essentially, if we knew that variable or that vector field ut of x, we would be able to just regress it with a neural net, vt of x theta, and that would help us somehow match the flow or match this vector field with just regression. And so people have framed it like this, where the loss of flow matching is essentially the square loss under all the potential time points, fictitious, again, fictitious time, and then sampling data generated according to this probability density path. And for those of you who know flow matching, you already know where I'm going. For those of you who don't, you would probably ask the question, this seems bogus, this doesn't really make sense, because I do not know ut of x. I have no idea what this is, and I don't know my probability density path. I just have a P0 and a P1, a noise distribution, data distribution. And so somehow we kind of need to come up with a way of engineering those, okay? And it's not easy. So in this same paper, Lippmann et al., who introduces flow matching, they actually propose this idea of conditional flow matching. And the idea is that it's really hard to know what the curve is between two distributions. But if you can simplify it and think about, here is a starting point, Z0, sampled from P0 at time 0, and Z1 sampled from P1 at time 1, then conditionally on knowing those input and output, or like starting point and ending point, we can just shoot up a straight line, okay? So it's a bit more complicated, because it's not just about moving points. It's also about thinking about it in terms of distribution. So you can write it like this. Basically, it's not about the conditional, it's not about the probability density path, but we have a probability density path condition on each pair of points, okay? And so we need to come up with a mean function and a variance function to kind of have this Gaussian deformation. And so here, for example, we can linearly interpolate in the mean between Z0 and Z1, and we can have a constant variance, for example. And you can play with different kind of variances. We will see examples later. So this is how you would define the probability density path in the conditional way. And based on that, you can specify the conditional vector field. So there are many vector fields that could satisfy this the same path. But there is one of them that's pretty simple, and it's somehow that one. The exact derivations do not matter, and I encourage you to read the flow matching paper to get a sense of how this works. And the idea is, now we have an actual way of regressing that vector field onto a neural network. And so the CFM loss, or conditional flow matching loss, consists in the square loss, mean square, or error, by basically sampling from random time points, by sampling from random pairs of points, and then matching that that deformation of like the following the line. Okay, so now this is pretty nice, and this has been used for generating images, and this has been pretty helpful. Then the key idea, I don't know what these slides are. keep moving forward. There is something wrong here. Okay. So the key interesting idea is with this, we need to understand how is conditional flow matching loss related to the real flow matching loss that we cared about. And what's very interesting is that if you somehow, you have this conditional vector field, so you can, in a sense, integrate and marginalize out this variable z to obtain a marginalized vector field. You can do the same thing for the probability density path. And with that specification, learning the conditional flow matching or learning the flow matching amounts to the same thing, which means that somehow those notions are compatible, and we have constructed a way to solve the flow matching problem by conditioning. And so that was what was proposed in this seminal flow matching paper. So, and in our work, we basically propose to extend this framework to multiple marginals, to multiple time points. And the idea is that we have multiple conditions measured at different time points. Let's see if I can make this. Oh, wow. Okay. No. Wait 1 second. I'm sorry. All right. So hopefully that will have solved my other issue. Okay. So we have those time points T0, T05, T07, T08, and T1. And for example, we have one condition that's measured at time 0, at time 07, and at time 1. And then we have another condition here that is measured at time 0, 0.5, 0.8, and 1. And so what we would like is to learn a vector field that somehow encompasses all those conditions and that merges all of those time points together. Okay. And we can basically follow the flow matching recipe and do that. So again, to specify the problem independently from all the biological background, I'm going to repeat it here. So we're given unpaired samples from multiple distributions at different time points that belong to different conditions. And we want to find a vector field that matches the flows for all the marginals and for all the conditions. Okay. And so again, thinking about this matrix of some conditions are missing at certain time, the idea is that if the model was able to truly learn something about the dynamics, it should be able to impute missing data decently. Okay. So is there any questions about the intention of how now the model is stated in terms of flow matching, or any question about flow matching?
Yeah, I had a question about P0, like in this slide, for example. Yes. So when you're explaining flow matching, conditional flow matching, it's like you kind of map some noise distribution onto a data distribution, right?
Mm-hmm.
And now how you're showing it here, it's like P0, T0 it's like kind of both in a way, right? It's like, it's a kind of a prior distribution, but that's made up of data, right? It's just, let's say, combining both conditions.
Yes.
So is that something you have to deal with, or is it something that's fine? Like you could express your noise distribution as still being in a empirical distribution over data.
Okay, so I'm really glad you're bringing that up. Basically, actually, so I've presented the flow matching as background in terms of noise to data, but flow matching has also been used in the setting of data to data. And so in that case, P0 does not need to be noise distribution. It can also be one type of data set, and P1 can be another type of data set. And in that case, you can use flow matching for like pairing data points, for example, or like style transfer, or if it's supervised or things like this. And so
I guess it's
P0 can be data as well. And,
okay.
And in that case, so that's why I'm clarifying this. I'm glad you're bringing this up. So in that slide right here, P0 is data, P1 is data, and all of those distributions are all data distributions. There is no noise or prior.
Right. So I guess that's like similar to, optimal transport, where you would transport one data set onto the other, let's say, right?
Yes. And so we're going to get into this a bit later on, actually. But the idea is that optimal transport is more meant for pairs of data points, but here you need something to learn the dynamics somehow.
Yeah, the evolution.
Yes.
And I guess if you needed to do some sort of variational inference, you would need prior with like a distribution, like a parametric distribution, or
yeah. We don't have any of that here. Yeah.
Okay.
So we will not be able to. We can still like somehow generate data, but there is no concept of prior in a sense. Yeah.
Okay. Thank you.
All right. So basically, we're trying to find like a vector field that matches this data in terms of dynamics. That's really what we're trying to do, and taking into account all of those conditions and being able to generalize across those missing pairs. When we want to apply that flow matching framework, or like slightly extend it such that we can solve that specific problem. So step by step, what we're going to do is first set up a conditioning variable that makes the problem solvable, like what we've seen in flow matching. Specify the conditional probability density paths, solve for the corresponding vector fields, and we can, and that will not be too hard, basically. And then we can extend for multiple conditions using classifier-free guidance. And I will also talk a bit about and then we can check out, and we can check out the predictions on the holdout data. I'm sorry for my screen. I don't know why this is happening today. So again, let's think of this in the cartoon way. We have those like 4 distributions, and we want to specify the conditioning variable for this problem to be solvable. So it's pretty easy. Somehow, for every time point, we just pick one of the samples, and this is a vector, right? Z is a vector formed by all of those. Again, and then the objective function, once we've specified our P of t and U of t, will be actually the same objective function. And it's a bit subtle because the Z is not the same, so you do need to derive back the equation, but it's pretty simple. It should be simple to convince yourself that when you solve this problem by adding more time points, you're essentially morally doing the same thing than you were with conditioning in the flow matching case. And so this is a valid objective function for multiple marginals. So this is not where there is much innovation somehow. What is interesting to think about is what are the probability density paths. So what we propose here is to use, because we have multiple time points, we can actually look into things that are non-trivial, for example, cubic splines. So given all the Z's, we will find a spline that fits through all of those points. We can also use piecewise linear function, but what's interesting is that the splines induce some kind of minimal curvature prior, and so can be pretty helpful in data that satisfy these properties. And we're thinking that gene expression is probably valid, or other processes. But we have a baseline in which we will use piecewise linear, which is a sensible thing to do. And then you need to define the variance function. And so the variance could be constant along the whole line, but somehow if the variance is constant and small, you are not giving yourself the opportunity for learning from other conditions. So I haven't talked yet about conditions, but this is why this variance is important, of this idea that the furthest away you are from the observed time point, the more variance you have. And that kind of helps you when you do the smoothing to kind of learn from other conditions as well. So this is how we specify the mean and the variance function. once we have the mean and the variance function, we can plug in that other formula I've shown you, and we have the u of t. And what's kind of interesting to think about is, all right, take a step back a little bit and think about how does this relate to conditional flow matching, okay? Because we've just proposed another framework, but what is its, are we actually doing more or less the same thing? In which sense? Are we generalizing it? And so what we've tried to explain is basically the following, that the design we used before is for experiments, but if you use a piecewise linear mean function, and if you use a constant variance, which is suboptimal, but it's kind of for understanding the relationship with this conditional flow matching. And then if instead of learning the vector field into a single neural net, you kind of use one neural net per pairwise marginal, so in between every time point, and you kind of dissociate them completely, then it's very interesting, because the the MMFM problem, so this multi-marginal flow matching problem, you can decompose it into k independent flow matching problems. And so the solution consists in just like kind of gluing the vector fields obtained from those individual problems. And what it means is that morally, wow, our objective is an extension of the flow matching objective. And it's a natural one, because under those specific assumptions, we recover flow matching. But somehow, when we allow the parameters to be shared across multiple time steps, we're actually able to somehow learn across time points. And by learning across conditions, we can also have some generalization. So that's why this our setting is a nice extension of flow matching. So,
hi.
yes.
I have two questions. So I have a very similar task than you're explaining, working on T cell with different time points and different condition. And indeed, what I went through intuitively was this piecewise linear modelization. And what I realized is that when your model are when your piecewise models are not perfectly trained, you'll just accumulate errors, because the prediction of T0 to T1 will have some error. But when you train your second model with the true T1 to T2, your second model will be applied on slightly out of distribution data. So is it something that you also observed? Is it something that you spend time to fix just on for this piecewise linear model, or did you really just solve it by this spline modelization?
That's kind of interesting. I mean, there are several things going on, right? There is first the concept of like error accumulating when you integrate, right? So you have this vector field, you have to integrate it, you do numerical integration. So at the end of the day, you always accumulate error through time. But what you're mentioning in your piecewise step is that it's different. It's not just error with the integration. It's actually errors by propagating like those models. And in a sense, the way that we do the training, basically, it's still a single model. So we don't need to wait for the output of a model to then put this back into the other one. So I don't think we would have that specific issue you're mentioning, but naturally, because those models are then used as integrators, they still accumulate errors, but not in the way that you just mentioned, I believe.
Yeah, that's my intuition as well. Because you compute some splines, so it looks like you're taking the global trajectory and not just the piecewise trajectory. And can you back up 2 slides, please? So the one with the when you explain the spline with the gamma as the argmin of a specific interval.
Yes.
Okay, this one, the cubic spline. Can you give us more insight on what is this gamma? I'm having a hard time to understand.
Yeah. So this is there is this concept of non-natural cubic spline, and it's basically a piecewise polynomial that gets through all the points but minimizes the curvature.
So what points is the points
So there is a typo in the slide. It's not such that zt equal gamma of tk.
Such that zt.
So
okay.
You have those. This z is a vector, z1, z2, z3, z4.
Okay.
You basically you find the spline that passes through all the polynomial that passes through all the points, piecewise polynomial, but that minimizes the curvature.
Okay, I think I get. Yeah, I get it. Okay, so in your framework, when you use OT, you start with a batch at T0. Each point, so Zi at T0 will be matched to point Z at T1 according to your OT minimap. And each point. Yep. Sorry.
I haven't talked about OT yet. I will, but basically right now the points are independent. You could sample, and that will not work that well, right? But like, or you can sample points at random. Q of Z is points from Z1, points from Z2, points from Z3, points from Z4, or at random in the samples we have, and then you can build mini batches out of this, and then you find the spline, and then you calculate the flow and you regress you calculate the vector field and you regress it. Yes. And so, I will talk about OT afterwards. I have a discussion.
Yeah, sure. Okay. And so that's what the argmin of this integral, it's really finding the polynomial that has some curvature and goes through all these points. Okay.
Yes.
Okay.
So I also have some question for this slide. So for the k, like for the intervals of the time step here, like you can see the tk plus 1 tk. So I think this is the predefined time steps, like from the data, right?
Yes.
Or how dense it is. For example, because when we usually do flow matching, it's from 0 to 1, but we would assume like the time is continuous. We just cut very small pieces, like very small time step. Each time we go to the next. But here, if the predefined time step is very large and it's also discrete, I'm not so sure whether this would lead to inconsistency with the standard flow matching, and I'm not so sure how that would make any difference.
Think about it as like this is flow matching, but you solve flow matching on different intervals that are stitched together, right? So when we solve things, it's still on the continuous scale. You sample time points anywhere, but these time points will end up in one of the segments. For example, tk to tk plus 1 or ti to ti plus 1. And you end up, so you sample things in there. So it's still continuous. It's just that somehow the way that you set up your curves for the flow matching, the way you set up the conditional density path, it depends on where you are. So this mu t is piecewise and the sigma t is piecewise, but otherwise it is you're still doing as in flow matching. It's solved on the continuous scale.
Oh, I see. So the standard flow matching is like from time 0 to time 1. Like you have a continuous, like for example, variance function. Like they are changing continuously as the time t. But here you just trunk the time steps as the predefined time interval from your data, which means that inside that interval, all the variants are the same. All the mean functions, they are kind of the same. Is that the case?
No. What I was saying is that basically T0 is 0, TK, TBK is 1, and there are just times in between somehow the curves that the definition of the density path and the definition of the vector field itself will be piecewise. And how you specify that is dictated by the data you have. But otherwise, it's exactly like in flow matching where things are solved in a continuous way.
Yeah, sure. Thanks.
All right, so let's see if now this lets me get back. All right. And so, okay, there are only 10 minutes left. So I will get through this quickly. So extending through multiple conditions is not something that's very difficult. Basically, for those of you who have worked on diffusion models or have worked on on flow matching, somehow adding multiple conditions should amount to just conditioning on that condition. That doesn't work that well, and it doesn't work that well because the neural net can struggle to learn across condition and share parameters. So what's usually done is this classifier-free guidance. And again, this is something we just adapted for the settings of this paper. So I will leave it for questions. And it's not that difficult to get to work. I will talk about the optimal transport because I had quite a few questions. So right now, what I've been talking about, I've been assuming that we can do independent sampling, meaning that we just take points at random for every time point within a single condition, for example. And we know that this does not work very well for the purpose of doing flow matching from data to data. So, for example, Alex Tong proposed this idea to use optimal transport for matching things. And if you apply this to this time point problem, you obtain the multi-marginal optimal transport problem, which is actually quite a tough problem. So I guess if there is anybody who knows how to compute this or like approximate this in better ways, I'd be happy to chat. But what we went for in that manuscript is basically if you assume that the cost is linear and Euclidean, which is not great, but that's fine. Then you end up with actually looking at the optimal coupling between immediate pairs of distributions. So for example, from time k at time k plus 1, you run optimal transport, and then this is exactly what the person, I can't see the names, I guess, on Google Meet, but the person who was this great question about how things work in practice. You would basically start from a mini batch of points at time 0, and you use the OT coupling for sampling points at time 1, and then points at time 2 and points at time 3. And this is how you build your trajectory. You build the splines on top of these pairings that you get from optimal transport on pairwise. So there are quite a few multi-marginal models that came up either around the time that we submitted or after. And there are two baselines that we looked into at the time of the paper. One of them was this splines plus OT work that basically consists in, it's a very simple method where to solve this problem, you compute OT across consecutive time points, which is what we did. You fit a spline with the time points, and then you use a spline to get the intermediate predictions. So this has two caveats, like if some times are missing, your spline might be really a bad approximation of the data. And also, it needs to be run separately for each condition. So morally, what we're doing is actually an amortized method to work across time points and conditions. And we will see that it helps working much better. And then there is another baseline called DMSB, Deep Multi-Marginal Momentum Schroedinger Bridge. And we really weren't able to get it to run. A lot of those methods based on diffusion or Schroedinger bridges are actually they take a while to compute, and they're very unstable, and so they're hard to work with. And we were hoping that using flow matching, we can have a more practical method, even if the theory might not be that nice. So, okay, there are a few minutes. I'm going to go through some of the results. So what we did some synthetic experiments in 2D, in which basically we can build a dynamical system in 2D. So you have those points moving in 2D space with time. You have different conditions. What changes is the location on the y-axis is changed with the condition, and then also the speed at which the y-axis will change is as a cosine. So it will look wavy, but we scale it up by the condition as well. And then we have different measurement scenario across conditions. So what the data looks like is like this, where you have those waves, and each color is a different condition. And basically, you can, so this is a very physical, I guess, model. And what you can see is that here we have some missing data in key places, and this is how we will evaluate the model. So this is heavily constructed to show that the model works and why other baselines could be performing terribly. And so the idea is that this is a generated data. This FSI method that looks like OT plus splines will work like this. So it will, because it cannot share from data across other conditions, it cannot understand that all of those conditions will look like waves. and it makes really erroneous predictions. But when we look at this with a neural net that can amortize and learn across conditions, it works much better. Okay. Yes.
Your question, your conditions here are ordered, right? It's not like a one-hot encoding of your condition.
In the neural net, I forgot how it's encoded. Yeah.
Okay, because this has a very huge echo with what I'm doing again. So because I would be interested to know the answer. Like, does it has learned some kind of ordering, or was the ordering already given in the model? Like,
yeah,
the condition token was ordered.
What I would imagine is that even if it's one-hot encoded, it still works.
Okay, but
and I think it's one-hot encoded in the experiment, but yeah, we need to look at the paper to be sure. That's what I would imagine is done. Yeah.
Okay. Thank you.
So then we can look at the results in two different metrics. One of them in mean square error, and one of them is error in distribution with the MMD. And then morally, I just want to show you the single cell data. So this is a subset of the million cells data set, but basically this is how we visualize the data with this SCVI and UMAP method. Each point is a cell here, and each color is a different drug. So you can see that there are many different kind of drugs. We cannot put a label here, but you see some localization of colors, and so that means that the model is picking up, and the cells that have the same drug have similar gene expression. You can look at the category, and so drugs that are similar will also be lookalike together. But then this is just to show you the data. This is not I'm not making any claims here. And then this is the time points. So time points look different in mixed cells because when those T cells go through activation, they will look different. And I will finish on so yeah, here I'm explaining why this will work, because the effect of the drug on the cells will look alike a lot, so we can do those imputations very likely, and we can look at the same metrics, and we can see that again. Sorry, I'm really lacking time here, but this is we're able to make those imputations when we can hold out one time point at a time and do this modeling. And so I will stop here so we can go for questions, but yes, and I will stop sharing. Thank you.
So before we open up the question section, let's take a moment to thank our speaker for the absolutely wonderful, enlightening presentation. Thank you, Romain, for coming out to the reading group and delivering such a nice presentation for everybody. So with that, if anybody has further questions on top of this very interactive session at the Multi-Omics reading group, please do so now.
I do want to mention that, Romain, there's a notice that you're running out of battery on your computer.
Yes, I know, and I don't have a charger, so this is. Yeah.
Okay, I thought it was me, so I went out and got my charger, but it's you. Okay, but okay, so we might lose you, I guess, in a moment, unless you have I don't know how much time left you have on your.
A few minutes, probably. Yeah.
Okay.
So I see a Philippe Sands, please. Yeah.
You're good, Dylan? Okay. I have other question, obviously, tons of them, but let's start with one. In my data, I have cell division, proliferation. I know I have proliferation. I can also measure proliferation. Have you thought about how to deal with this optimal transport coupling to take into account that you have some clusters that can just grow and some that can collapse, such that you don't have mapping between that are just fake mapping coming from the fact that the real optimal transport is an unbalanced optimal transport?
Yeah. I mean, yeah. So I guess the idea is that you would use unbalanced optimal transport, but I don't know how it gets back into like the flow matching and how to make it principled, and what are the properties of what you learn under usage of unbalanced optimal transport, right? So when you use OT between pairs of time points, in our case, we don't have actually the McCann interpolant anymore. We don't have this interpretation, and I don't know what it would take to kind of bring it back. But in what
McCann? Okay, yeah, I see.
So when you do between flow matching between pairs of time points, I guess, exclusively between two distributions, and if you use OT for aligning the points, in principle, your flow matching learns how to fit the dynamical solution to the dynamical OT.
Yeah, okay.
And so I don't know how much this is how important this is, and in our work we cannot prove that, and I don't know if it's critical or not. And so one key question to understand whether you can do unbalanced OT and flow matching would be to try to figure out do you still get a similar, like dynamic or like nice property when you have only two time. You can start with two, right, and then with unbalanced OT, what do you get, and then try to bring that into another setting. But so yeah, this is, and no, it's an interesting question because the T cells proliferate here as well, right? So especially at 72 hours, where you can see huge differences in cell numbers, and this is not something that we've really looked into. I mean, we've looked into the data and we know that what you're saying is exactly right, but we haven't looked into how it affects the modeling, and that probably should be interesting.
Okay. Thank you. I let the floor for the question and come back if there's none.
Do we have any additional questions from the audience?
Maybe any insight from the future work slides? I see you just skip it.
Oh, from future work, yeah. I mean, yeah. So we already are mentioning this actually. I think there are like different options for figuring out this kind of work. So either use flow matching and you build some practical like implementation of flow matching, and I think this is what we did. So in a sense it's very humble because this is just a solution that's practical and that seems to do something decent. What's interesting to think about is what would be a nice theoretical way of doing that. And so maybe some of the folks on stochastic interpolant or some of the folks on this Schroedinger multi-momentum, like multi-marginal Schroedinger bridge, I figured out what in theory would be really nice to build, but the algorithms don't really scale in a sense. And so it would be nice to think about intermediate like things. So that would be really great. I would love to think also about how to tie those. So with the vector field, in principle, you are solving some kind of differential equation. And I would love to think about how to tie this more to systems biology, or thinking about whether there is a way of like smooth regularizing the network, maybe for like making some of the changes interpretable, and really reasoning about what are stable points of the dynamical system, or like thinking about that in a sense. I would love to also think about how to tie those these models that are meant in cell state, like SCVI space or PCA space or whatever low dimensional space you're working with, and trying to understand how can we extract biological insights from that. So it would be really nice if we can have those models work on the space of gene expression. And it's just super unfortunate because neither optimal transport nor diffusion models nor flow matching work on, to my knowledge, work on spaces that with that high dimensionality. So those are the kind of questions that I'm trying to understand. And the reason to go to gene expression again would be to basically build causal model that can, or like model that are at least putatively causal, and that can help you nominate, the drug maybe acts on those gene processes at a given time point, and then the rest is kind of like conserved and unrolls through time. So kind of try to figure out what is direct and what is more indirect through the time measurements and the perturbations. I think are like also directions I'd be interested in exploring. I just don't know how much is feasible with the quality of the data that we have, the number of time points, and computationally what's tractable as well.
Thanks.
Okay. Any more questions? If we do not, that finalizes the recording. Yeah.
I do, but I have a question. So sorry. So you present multi-marginal, not necessarily OT, but multi-marginal flow matching, I guess, because you have one marginal distribution per time point for which you have data, right? And I couldn't help to think about like also multi-marginal OT being a solution to the barycenter problem, right? So definitely, like you have two distributions, two marginals. Let's say you could kind of find like the interpolant, like middle distribution, right? Like the centroid, let's say. So that's the barycenter. It turns out that the solution to the barycenter problem is a type of multi-marginal OT problem, right? So, and we kind of see it because you could think of it like at t0 and the last t, all the other marginals being like different types of barycenters in a way, right? It's like you're interpolating along one spectrum in a sense, right? But I wonder if there's a way to extend like this barycenter multi-marginal problem if you learn barycenters across your different conditions as well, right? Because now you're connecting like pairwise. And so if you're connecting pairwise, you have to do it. You have to have different pairs for different conditions, right? But is there a way to like harmonize everything into like maybe one kind of almost like singular representation? It's maybe not the most precise, but it could give you like maybe other axes of variations across conditions.
Yes, I mean, some of the work that we're interested in, it's not going to exactly answer your question, but it's the related thoughts that we gave was about, right, what would be the right way of doing the optimal transport to recover the McCann interpolant. And so I guess if you solve the multi-marginal OT problem, then somehow I think you probably can tie it back to the McCann interpolant. And the McCann interpolant is related to this barycenter and, you know, this process. But because the McCann interpolant is basically like taking the Monge map and like pushing it and scaling it in a certain way. And I think that I think it's related to the barycenter problem as well. I'm not sure. But basically, we were interested in formalizing that more general problem. And you need to think about the cost function that link all the data points together, right? So for example, for every data point, like what I was talking about, Z1, etc., you would kind of score it in a way that would be your cost. And the cost could be, for example, the curvature of when you minimize the curvature of the spline, it could be like how long your path is or how curvy your path is, and you can come up with a number. And I think the problem is that none of this is like retractable, right? So I think either you need to think about how to break down like this, and maybe it could be that there is a nice formulation of optimal transport in a multi-marginal way when your cost is some kind of approximate energy. So maybe not pairwise, maybe it's not too great, but maybe you can do approximate with some finite differences, some local energy, local curvature. And there is a way that somehow this kind of like tantamounts to some much simpler problem than solving across the whole time series. And that then by doing that, you would get better pairing, because right now the pairings, we know we claim that one of the results in the experiment is that spline works better than piecewise linear for the guide of the change in means. But somehow our OT map is piecewise linear because somehow we don't take into account the curvature of the pairing between, we don't take into account the curving of that line joining 3 points to kind of bias the solution of the coupling to be less curved in a way, you know. So there is these things are solved independently. And so I think it would be great to try to think how to bring this curvature idea into the OT as well. And then maybe then you can tie it back to more like this idea of barycenter and this idea of McCann interpolant. And this is kind of the piece that's missing there.
Yeah, nice. That'd be really cool.
But I don't know enough about this whole literature and how it translates to multiple marginals. I mean, at this point, I would love to think about it more, but
there's also like factored OT. It's not quite the same thing, right? But it's like if you think of your transport plan as being you could decompose it into like different bases, right? What's been shown is that then you could also use it to identify how connections between different marginals are part of a larger transport plan. But anyways,
that's interesting.
I'm not knowledgeable enough to say that I know with confidence that that's like a promising direction.
Yes, thank you.
Do we have any additional questions from anybody in the call? Please do so now.
Okay, I do. How did you try to observe the dynamics in the latent space rather than cell states?
To me, that's the same thing. What do you mean?
So what I mean is that I personally want to plot the trajectories that I have found and describe each cell not by and describe each cell by the vector or the matrix of the different states over time, and observe that matrix in a latent space, rather than plotting each individual point, because if you plot each individual point in UMAP or whatever, you won't have the you do not have the trajectories that you have found through your method.
I guess, but the okay, what I'm confused about is the method doesn't really compute.
So the method generate trajectories for each starting step.
Yeah.
Each starting step. So instead, if we work with the tabular data, each observation is a cell, different time, and so on. Then in computer UMAP, each point would be a cell at different time. But if we do the flow matching approach, then each point would be the trajectory of an original cell at time 0. So all the other cell would be just thrown out, right? Because the trajectory will be estimated. So now what I would like to do is to project this trajectory in a latent representation, because similarly to your example, where you have this kind of curves that are in fact ordered, you have one curve that is over another one and so on. It looks like there's some, I don't know, ballistic embedding, for instance, the force and the angle at the origin that could be enough to describe your dynamics, right? In your toy example. And so similarly for me, but in higher dimensional data, because we're talking about transcriptomic data, I would like to observe the dynamics in the latent space. And so I don't know if it's a question that you've addressed already.
So you mean like embedding trajectories, basically?
Yeah, exactly. And it has some usages, because if you have some drugs that have similar impact but just with different intensities, what you expect is that the cell for the dynamics for the dynamics in the embedding space will be ordered by the strength of the administrated drug.
Right? Yeah. So I mean, we have basically for the larger data sets, I've been thinking about other type of models we could use for like data exploration. The flow matching model is not necessarily the best for like data exploration, but it solved the task we came up with like really nicely. And so in that case, well, I mean, some kind of ideas that I had was about like this idea of figuring out basically it's kind of like embedding meta trajectories. So like whenever with OT you can get like, you know, some trajectories in a sense, and then you learn embeddings of those trajectories as a whole, and then you can understand each point becomes not just a cell, but like a trajectory made up of several cells. And then you can look at the latent variables and then you can see how your chemicals are separating or converging on like the latent space somehow in that meta space of trajectory. So you can build hierarchical models like this.
Okay. I'd be happy to discuss more that with you. I don't know if you're still here, Romain. Okay. Yes, I would be happy to discuss that more with you.
Nice.
Later.
Just want to ask a quick question to jump in the discussion. Like, do you think it's possible to do single cell level like emergence like modeling? Because like we only get a distribution of cells. We don't know the exactly mapping. So the OT is just some mathematical framework to allow us to find the mapping between cells, but it might not be real mappings.
Yeah, and so what is yes, I mean, so I agree with you that the OT approximates something that we can discuss, but and I'm happy to discuss that and the limitations. But what is exactly your question?
So you mentioned like in your question and answering earlier, like you mentioned that you are thinking about something to model the single cell level developing processes. Like for the exact cell, I'm not so sure if I understand correctly.
I see. Well, so yeah, the model is able, like the flow matching model in principle is supposed to be able to take a particle and push it through, in time, and you're supposed to be able to see which particle you arrive. And again, it's very guided by the optimal transport, right? So any bias you have in your optimal transport, or anything you're trying to or any debiasing done because of the coupling will appear also in that like push forward map. It's kind of interesting. So we looked carefully. Actually, this is something we did in the data analysis part of like, you take this T-cell data, you do OT between one time point and another time point, and then you try to see like what variance is it matching on, right? And I mean, it's matching on whatever variance is present in the embedding, okay? But like specifically, we wanted to know other biological processes that it's matching, and does it make sense or does it not make sense? And so in this setting, what we found is, for example, it matches across cell cycles. So that means that if T cells are in a certain cell cycle, at the other time point, they will be matched to a cell in the same cell cycle. If you think about this, it's both like a blessing and it's also really weird. It's a blessing because it makes sense that cell cycle is a variant that's present nevertheless, and so you probably want to match it because it's easier than for the predictor to really focus on the effect of the drugs and not about what the cell is actually going through and how it ends up in a different cell cycle. But if you think about it biologically, cells are cycling. They're always cycling. So the same cell, if you know its cell cycle at a given time point, there is no reason it will be the same cycle at another phase at this next time point. But it's still helpful. So that's like one setting. The other kind of thing is that maybe is more credible is every cell has a different strength of activation. And when you put them through those drugs, so the drugs will dampen activation, or they will promote activation, or they will like change it in some capacity. But either way, if you look at some marker genes for activation, there is always a distribution. Some cells are at the top, some cells are at the bottom. And so those cells come from different patients. They could just be also like have different sensitivities based on their history. And you recover that in the OT. So cells with high activation or match with cells with higher activation. And thinking about this biologically, maybe it kind of makes sense. If the heterogeneity has nothing to do with the drug, then it's nice to be able to match those things. But yeah, so I think it's kind of an interesting setup where I think matching this part of the variance is helpful. So that's why I think it's probably nice to do optimal transport and then matching. But is it completely a biophysical model of what's happening? Absolutely not.
Thanks.
All right, very good. Any additional questions? Like, I'm sure there's plenty, but we've gone 20 minutes over time, so I think that in the interest of time, Professor Romain, if you are very much interested in continuing the discussion, I'm pretty sure you'll get plenty of emails. But as far as the recording goes, that ends the 34th session of the Multi-Omics Reading Group. Thank you, everybody, for coming.